I'm happy to be joined today by Dr. Christian Dittrich. Dr. Dittrich is head of the 3rd Medical Department at the Center for Oncology and Hematology at Kaiser Franz Josef-Spital in Vienna, Austria. He is also director of the Ludwig Boltzmann Institute for Applied Cancer Research. His talk is Clinical Achievements Reached by Interfering with the IGF-1R, PI3K, PTEN, AKT, mTOR axis. Thank you for joining us. Would you give us an overview of the clinical achievements reached by interfering with these pathways?
Let me start with the success story of inhibiting by the mTOR. The first study which was run inhibiting mTOR was in renal cell cancer in unpretreated patients, and this was a trial comparing temsirolimus at that time with the standard treatment interferon alpha, and there was a third arm, a combination of interferon alpha with temsirolimus. The paper was published by Hades, and the data really brought a very significant and important decrease of the hazard ratio, the risk of death, and in addition also the same was shown for the progression-free survival. What was shown for the single-agent comparison could not be demonstrated for the combination of the temsirolimus with interferon, and this was due to a loss of dose density analyzed later on because of unforeseen toxicity, which let us overthink the situation that you have to do always proper prior combinatorial treatments, phase 1s, before you're going to combine such new agents. So this led to the registration of the first candidate of this pathway by first FDA, later on by the European Health Agency, EMA. And this was the start of one of those candidates to be registered. And the second story was the treatment of, again, renal cell cancer patients, but now it was patients already previously treated with an anti-angiogenic therapy candidate, which at that time have been registered. And for the first-line treatment, it was sunitinib, and for the second, it was sorafenib. And the trial with everolimus was randomizing patients relapsing or refractory to one of these agents versus a control. And the trial targeted as primary goal a progression-free survival advantage, and it made it with an almost 70% reduction of hazard ratio for progression-free survival. So after these two absolute stories of success, people thought over whether there were, let's say, signs of clinical activity comparable to what one was used from conventional therapies, and one found out that the response rates were rather low in the level of below 10%, only besides in a single situation, which was lymphoma, indolent Hodgkin lymphoma, and in addition, or specifically, it was a mantle cell lymphoma. And so there was another trial run, which again became a big success, published by Hess and co-workers, and they compared temsirolimus in a higher dosage, starting with 175 milligrams per meter square, and weekly later on 75, versus a lower dosed with a weekly dosage of 25, versus a control. And what was found out, this was done in previously treated and refractory mantle cell lymphoma. They got a highly significant and impressive reduction of the rate of progressive disease the trial was not able to demonstrate any survival benefit. But what was found out in addition, there was also a superiority of the higher dose level versus the lower dose level. So this, for the first time in this kinds of trial, a dose effect also could be proven. There are two more moments I'd like to mention. Everolimus was again a drug which was tested in neuroendocrine pancreatic cancer, and this was compared with placebo at that time, and it was reached in almost, again, 70% reduction of the hazard ratio to progress versus the placebo. And when the interim analysis was done, the data monitoring committee allowed to open the trial. So there was a crossover which later on inhibited, of course, to find out any potential survival benefit, but it was a great story. And maybe I should add a last situation, the one of the orphan indication of an subependymal giant cell astrocytoma with tuberous sclerosis, which is really a rare situation, but is a situation where there is a genetic defect which is known that there is a connection with the tuberous sclerosis and there with mTOR complex. And for patients who cannot be operated on, the treatment with a drug yielded at least two interesting remissions. So this is the, let's say, the success story concerning the mTOR inhibitors. If we go up the pathway up to the IGF-1R inhibitor, for none of the other nodes of the axis, neither for AKT, not for PI3 kinase, and not for insulin-like growth factor 1 receptor, there was a similar situation proven until now. There have been a demonstration that the mode of mechanism, the proof could be delivered, and the same was true for even getting some remissions, which can be taken as a sign of activity, but in none of the other studies so far any clinical enough impressive results could be demonstrated, not even for prospective randomized phase 3 trials, which were so far run for the insulin-like growth factor receptor inhibitors. There are two classes. The antibodies were the more advanced ones, and although with some of them there were some even clinically complete remissions, specifically in Ewing sarcoma patients, when a trial with non-small cell lung cancer patients was comparing a standard treatment with cabutaxel plus minus, in this case, figitumumab was done. After an interim analysis, the study was stopped due to futility reason. And another trial with the same compound also which had achieved already the phase 3 situation, where patients were treated either with elatinib single agent therapy or with elatinib plus figitumumab. The study was also stopped due to futility. Another compound which also was rather far developed with regard to IGFR1 inhibition, a Merck compound which was tested in colorectal cancer in a phase 2-3 trial, also was stopped earlier because of results which did not meet the endpoint at all. So what was seen with all the compounds tested so far, and there is really a plethora of substances, remissions have been reached in individual cases, but no further drug made it so far on a single agent in the phase 3 and came out and was determined to be superior to anything else.
So what are the next steps for your research?
There is certainly first the question of asking the right questions to find out what are the reasons why we are not more successful in doing what we were doing so far. And one of the answers would certainly be, did we do the right patient selection? One elementary question would also be to work on an improvement to find out the right biomarkers at the right time. Many times it is only that biomarkers are found to be standardized enough and validated enough at the very end of a developmental process. Whereas it would be very necessary to have these tools already available very early on. And one way of doing that could be to do preclinical studies or phase 0 studies where one has less the aim of doing already any clinical activity work for patients with the intention to tumor remission, but where one tries to optimize preclinical assays and get them get their proof of mechanism to have them ready once one is starting phase 1. And this would be one way one could ameliorate the actual situation. Another would be to get the actual information on what pathway actually is a driving force of a specific tumor. And for that we have to get actual biologic material, means tumor biopsies. So I think for so-called targeted therapy trials, we have to ask patients to allow us to get their tumor material in order to be able to develop drugs rationally. And I'm also convinced, and I got convinced also from discussions with patients, that they would be more than willing to do so once they got informed how we are dealing with these drugs. So we many times have more to convince ethical committees and other committees to accept also repetitive tumor biopsying, which of course will only be feasible up to a certain level. In addition, one should use more and more non-invasive methods to get actual information whether a drug is active. This can be done by radioactive labeled radio imaging, but also by not radioactive imaging methodologies. So far, the radio imaging is much more standardized, although there is still room to improve, whereas there are many other methods like DC MRI, which into my eyes have not reached the level necessary of standardization as of yet. So we have to improve also on this side. One other way certainly would be to combine therapies. As in all other areas of oncology, we are treating usually tumors not with a single agent, and the reason is quite evident. Tumors are heterogeneous, and for this reason it may be chance that we are just in front of a monoclonal disease, which is a rare event. And another important reason is, let's say, the redundancy of regulation of our homeostasis to keep us alive, but this also keeps tumors alive to a certain extent. And one of the results of this redundancy is once tumors cannot be deteriorated enough, resistance is developing. And the resistance may be resistance of the same pathway, but it may also be by the use of another pathway. And so, as in previous successful anti-tumoral therapies, we also will have to combine therapy with the targeted drugs attacking the PI3 kinase axis with other drugs, either with other drugs of the same axis to do some kind of double hit of the same axis, or to inhibit an axis which may be another way to overcome the inhibition which was created by the first drug. For instance, as an example, PI3 kinase axis may be overcome, or resistance may be overcome by an inhibitor of the MAP kinase axis, and MEK inhibitors are actually certainly drugs which are of special interest to be combined with drugs of the PI3 kinase axis. We also learned from breast cancer, for instance, HER2 positive breast cancer, which got resistant, that by attacking the PI3 kinase axis either afterwards simultaneously, that this could overcome the resistance. And there are very interesting trials which started already proving that. For instance, one work from the MD Anderson group combining trastuzumab with everolimus in a phase 1 trial, where they got in previously trastuzumab-resistant patients almost 14% partial remissions. And there are other examples, for instance, combining members of the PI3 kinase axis like everolimus with anti-hormonal treatment as another axis, as the estrogen axis, if you want. And there is this wonderful example of the Bolero trial from Jose Baselga, who showed in a very convincing way that combining everolimus to exemestane was clinically very much superior to the single agent therapy. So there are already right now good examples for combining, and one can imagine if doing the permutation of all the steps within the different axes available, and all the many drugs already available so far, and all those to be still discovered and still created, we would have an enormous amount to work on and to deal with. And this also may be a difficulty in another side, because we have to do the right choice, because we won't have enough patients to do all the permutational possible testing available. So we have to cleverly select what trials with what drugs to be done and what not to be done. Sometimes what not to be done may be even more important, to have enough power to focus on the most relevant.
So thank you very, very much for joining us, Dr. Dittrich.